I think we've had sort of today sort of a mix and match of quite technical scientific presentations and then some practical information and application which we can all take home with us. So I think this session is not any different. I think the next presentation hopefully is very thought-provoking. If we can just change the CV there we go. So Dr. Henny Groenewald from Biosafety, Gerrit and I had the privilege of seeing one of his presentations at another occasion, and it was really extremely thought-provoking and informative. So we're very, very privileged and look forward to his presentation. Thank you.
Thank you. Thanks. It sounds like I'm on the speakers. So hi everybody, Henny Groenewald. This morning when I flew up here, I was thinking about what messages do I want to convey, because I think when you talk to people, it must, there must be a message. I always dream that you have a braai next Saturday, and when you stand around the braai, you're going to say, yeah, I heard this guy say something very interesting, and then you start a conversation from there. That's my dream. And I want you to leave you with something, otherwise I'm wasting your time. So usually when I speak to regulators, it's clear we're trying to convince them to be science-based, whatever. It's clear what I want to do there. When you talk to people that do research and development, this is where I spend most of my life, so I know what to talk about there. But you are a mixed bunch. So in the end of the day, I thought it's probably something about innovation. So I'm going to talk about a very small part of innovation. I'm going to try and make it relevant to the people specifically in the sunflower industry as well. I'm going to talk about genetic innovation. And hopefully I'm going to talk about lots of different things which I will emphasize a little bit later, and why I need to talk about technology, and I have to talk about regulation, God forbid, and I have to talk about, all of these different things, but you need all of these things to be innovative in this space. And I think one of the biggest messages for me is, innovation in such a complex environment, we need a lot of different inputs. We need people with enthusiasm and energy to make inputs from different places. You know different people than I do. You can have an influence somewhere. In the end of the day, if our dream in this environment of, say, sunflower genetic innovation, if we want to have a product in the end of the day, we will have to influence a lot of different people. We have to motivate a lot of different people. We need funds, for example, to do specific research and development, all those type of things. And hopefully this is the message with which I'm going to leave you. But to get there, I'm going to run around between a lot of different talks, and where I'm going to, or topics, I should say. And where I'm going to start off is just quickly context. Biosafety South Africa, this is where I work for, who am I, what am I doing here? So in bioinnovation, so my background also is plant molecular biology and physiology, so I worked in the sugar industry a lot and some in the wine industry. But in the end of the day, anything in biotechnology and innovation, biotechnology, the word tells you, it's not just about the biology and the information, it's about developing some product, some technology that we can use, which is based on the information or the resources that we get from biology, from nature, in other words. But specifically in the regulatory technologies, and you will see I refer a lot to GMOs, because this is one of the things I'm going to do when I talk about induced genetic variation, when I get there, it's a regulated environment. It's, fairly complicated technology, so you need to invest in systems to realize your dream of developing products in that space. And some of those barriers are things like cost, expertise, compliance, and acceptance. And I'm going to touch on all of these different things. So what Biosafety South Africa tries to do is to facilitate this process in bio innovation to ensure that we have a shortcut through these things. So we are a service platform funded by the National Department of Science and Innovation. So, and then you will hear me talk a lot about sustainability. So what does sustainability in this context mean? We are called Biosafety South Africa because in the history, biosafety, if you talk about the biosafety of a GM product, for example, it focuses on the environmental and the food and feed safety issues. And again, I will explain all of these things in a little bit more details later. But then also there's these viability issues. So when you come from a research development side, you always focus on the technological side. I'm going to talk about genome editing today, and we usually, as researchers, get very excited if you have a new tool to play with and you can make new things. But that's only one aspect. If you want to put a product in the market, you only have a product if somebody is interested in it. So you have all of these issues about acceptance, for example. Your tools, in other words, must also be able to develop that tool. And some of the things that I'm going to highlight, it's one thing to get all excited about new technology, but if we cannot really harness that technology in the form of a product or go through the process to have a product in the end of the day, the technology is useless. Doesn't matter how sexy it is and how excited I get about it. The social-political aspects there is more in the social is the acceptance. If you talk about GMOs, for example, there will quickly be a lot of people that starts talking about, if you just do a GMO Google search, you will find out that people are concerned about the safety issues. Is this food going to give me cancer? Is it going to kill all the bees and all those type of things that you hear? So how do we verify that these things are safe? This is what we talk about. So if you have a product in the end of the day that's both safe and sustainable or viable, we define it as sustainable. And this is the focus and the backbone of all the work that we do. So we do not do product development per se, but we ensure the sustainability. So if somebody develops a product, for example, very early stages, people at universities or some of the research councils, for example, we will help them register their facilities because it's part of the regulatory system without charging them fees because we use taxpayers' money to facilitate this whole process. Or we can go to the development stage where people have, for example, have to test. Say, for example, they put the BT gene into maize. What does BT do? It actually kills insects. So what is to say that if I put this BT maize in my field, I'm not going to kill all the other insects as well, including the bees and the butterflies and all the other things that we like and that is important in the ecosystem. So we will fund, for example, that research. We will help with the technical side. We do the risk analysis, which is the basis. So the basis of sustainability then also, very importantly, is policy. We will not be able in South Africa, for example, since the mid-1990s, we have commercialized GM crops in South Africa. We would not have been able to do that if we didn't have a regulatory framework in place. We wouldn't have a regulatory framework if we didn't have a policy that supports broadly, bio-innovation in this space. But at the end of the day, and one of the clashes that we have at the moment, for example, with the regulators is that, okay, now we have regulations, we have these policies, but you must make decisions in this context based on science. You can't now just go and look at acts and do interpretations, that's based on words and just the legal aspects, for example. This is one of the things that we're trying to sort out, is that in the end of the day, when I make a claim about the safety, for example, I can submit scientific data to show you A, B, C, and D. This is why I say it's a safe product. And then genetic variation. Why do we talk about genetic variation? It's because genetic variation is the basis of, and I now use words like phenotypic variation. It basically says if you have different sunflower cultivars, whatever the reason is you selected a specific one because it fits in your environment, because it needs a little bit less nutrients, whatever. I know nothing about sunflower farming. Unfortunately, I actually grew up between Klerksdorp and Ventersdorp, and my dad used to plant sunflowers, but I just played, when they put it into the big trailers. The point is the cultivars that you select, why do they have those particular traits? It's because of the genetics. Why do we have breeding programs? It's basically to put the genetics, the traits that we need through combinations of selecting a dad and a mum. Hopefully, the offspring will then have some combination of those traits, and this is how you then improve cultivars over time. So, genetic variation, I also talk specifically about induced variation. So this can be a gray line in this work. In biology, you should realize everything is gray. There are not black and white answers in biology. Biological organism can one day behave a certain way, and if the environment changes, it will behave in a different way. So it's a complex system in which we are operating. In any case, genetic variation, it's a natural process. I have those two pictures there of two different types of moths. It's the peppered moth in Europe, but you see there's two different types or different phenotypes. The one is light and pepper colored, and the other one is dark gray. I have them on the two different backgrounds because what will happen if a bird passes at the top, those two in a dark environment, it will see the light-colored one and it will eat the light-colored one. So what is the result in nature? More of the dark ones survive. In other words, you're going to have a bigger chance of a dark male and a female meet each other, and they will have dark offspring. And this is how evolution happens over time. It's not, some people think evolution is this thing where you stand there and say, I have to adapt because, there's no dark environment or whatever. It doesn't, evolution doesn't work at your generation's level. If there's selective pressure, like I just tried to explain, you will see this precipitate in, the subsequent generations. And then we started to discover how genetics work. And of course, if we're only going to talk about plants, it's going to be very boring. So I brought in dogs. Again, I'm not a dog breeder, but I love dogs. And the variations there. I just had to select two. This is actually one of the things where I spend most of my time when I prepare for a slide. It's not the technical stuff, but it's just decide which dog pictures I should use. But yeah, we breed for certain levels. Dogs I use as an example as well because we just do it for fun, basically. We love our dogs, obviously. But there's variation, so we can breed. So what is breeding actually? We're just using that natural variation, but now we say, we're not just going to leave you, to have a free-for-all out there and we see where the dog population is going, which there's actually a nice example. Sorry, I'm jumping in here. But if you go to an environment where there's a lot of dogs running around, you can see the genetics drift in a certain way. And then you see dogs with a particular trait. But if you want this little thing at the bottom, obviously you must keep it away from the burbu or the other dogs. And then breeding, it evolved. Then we started what we call in vitro breeding, where you could actually, that ruby grapefruit, that was actually part of a program where they used high energy radiation to induce mutations. So now we understood, but listen, the genetics is the basis of what we see in nature, and those changes are actually because of different, sorry, my son is trying to phone me. Those variations then can either be good or bad, and we can select them then in a breeding program. But then we realized that those mutations, the changes in the nucleotide sequences that, can be induced, or it comes naturally, but we can also induce them, and this is when we ended up. The banana, I should talk a little bit quicker about these things, but banana is a triploid. So that is basically just a cross in which you change completely the number of chromosomes in there. And because there's now suddenly 3 chromosomes for every what we have, usually called pairs, but now there's a triplet. And when you have meiotic division, that can actually not happen because that mechanisms, if you have only 2, you can separate them into 2 cells, which will give you then your gametes. But now it can't happen. So what's the eventual result of this? I can't have seeds. And that's why a banana that we eat and that we know does not have seeds because it cannot, on a biological level, form those seeds. Then we started with genetic modification. 2 examples I have there is maize, BT, herbicide tolerance, all those type of things we know. The only animal that's only been approved in the US and Canada and a few places is genetically induced, modified salmon. Those are the same age, so the top one just expresses the growth hormone of the fish at a higher concentration. So you increase the growth rate. And now, today, I'm going to talk specifically about genetic engineering, and the products that I've got there I'm going to talk about later. So the point I want to make is this whole range of genetic variation and the ways in which we can manage and induce this. And there's lots of complexity here now if you start talking about how am I going to do this, what is the best technology to use that will put my industry in a competitive advantage. And if it's regulated, it gets even more complicated. So I already said I'm going to talk about technology, products, and impact. Impact is what we're actually looking for. If I'm a sunflower breeder and I can't make new cultivars that the farmers can make money with, I'm not doing my work, basically. So the genetics, all these things are interesting, and they're a means to an end, but it's about that impact. And like I said, unfortunately, I have to talk about regulation because these technologies are regulated. Sorry, I'm jumping around. So I will jump a little bit around these because I cannot really tease them apart, but I will try and keep you, organized as I go through it. So this is exactly what I said earlier. What I want to do now is just imagine now you can, see into that cell and you can actually see what's happening on the chromosomes. And it's a very simplistic example just to show you that I've got my original cell at the top, very traditionally. I've got a mum's chromosome is the pink one and dad's is the blue one. Then through things that can happen like sun irradiation, this is why you get skin cancer if you sit in the sun too much. It can be actually the molecular mechanisms. Remember, every time, each and every one that's sitting here, at some stage you were one single cell. So everything you are is from that original cell. So that cell has to divide and the DNA has to be duplicated every time. So there's molecular machinery that's going on and turning like mad your whole life. And sometimes they make errors. If those errors are incorporated into your gametes, your sperm or your egg cell, that changes are actually transferred to your kids. And this is again why then in the end of the day, and you're a combination of your parents obviously, but this is why you can see. But kids can also have completely new traits because of these induced genetic variation. So these can be single mutations, it can be rearrangements, it can be duplications of deletions, and then in vitro breeding, the one that I showed you where we now started to do irradiation. So this is why I've got this picture here. This is a way to induce these changes in the DNA. But the point I'm trying to make, you see here, this is what happens naturally. This is what's happening when we do in vitro breeding. This is what happens when we do genetic modification. So genetic modification, you take your original cell and it's 99.99% the same, except that you went and you fetched another gene that codes for a specific trait that you want. For example, insect resistance, the BT gene that's in maize gives you insect resistance. So we got that from a bacterium. So that gene's never been part of maize before. So this is one of the disadvantages of breeding, is obviously breeding depends, per definition, on sexual compatibility. So one of the products that's going around the world now is the drought-tolerant soybean with a gene from sunflower, actually. You can't go and cross sunflowers and soybean to get that one gene over, but we can do it with genetic engineering. So there's then now my new purple gene in there. If I talk about genome editing, you use specific tools, which I will very briefly look at, to induce a mutation as well. And then you will see now, suddenly I bring in a new term here, which I call SDN1 and 2, and there's a 3. So the only thing you have to remember, they are different, and I will talk very briefly about it a little bit later. But what is the reason behind this very long-winded slide of mine? Is I want to show you that some of these mutations, we refer to them as point mutations or small deletions and things like that. It doesn't matter if it happens naturally through these old technologies that we've used since the 1940s, literally, or genome editing. I can induce, if you look at the cell, and remember, I can regenerate, like I explained earlier, the single cell now into a whole new organism. There's no difference. When I only look at the organism at the end, I will not be able to distinguish between these three. So this is a very important point in the discussion that we will have today. The other point I just quickly want to make here is that if you do breeding or this, when they did the irradiation and those in vitro things, you literally have a target of millions of cells and you just, put a lot of irradiation on them and hopefully something will come out and you have to go through this long process of planting them in the field and selecting and on, all this breeding stuff. And so you only select your traits reactively because you cannot say, geez, it would be nice now to have an insect resistant, and then there's not a way that suddenly it will appear. You have to try and breed and see if there are plants that are resistant, and long story. But now with genome editing, I can do it targeted. I can actually design traits now. If I understand, there's always an if and a but. If I understand what gene gives which phenotypes, I can change those things now, because remember, we've got a long history of reactively seeing if there's a mutation in this gene, this happens. So this is how we, kind of sort out this whole complicated DNA stuff and then do it. So one of the big advantages in genome editing is that it's more specific, and I'm going to explain to you now why. So this is the only technical slide, hopefully, that I'm going to show you, but I try to keep it as simple as possible. So you've probably heard people talk about CRISPR. So let's first start there. I talk about genome editing, and I've spoken about genetic engineering. No, I have not. I said genetic modification. So the Americans actually like the word genetic engineering, which is a broad term. That's just what the word says, genetic engineering, anything that I change and do and things. We talk about genetic modification, and you will see why this is important, because later we're going to say genetically modified organisms are regulated. But now I've start talking about genetic gene editing. So I'll get to the question then later, are genome-edited products GMOs or not? And should they be regulated? So this is where we want to get to the discussion. So when I make a GMO, usually what happens, and this is the way that we want to define the term, is to say, I go to a bacterium and I put it into a plant, and now that plant's got that new trait. For example, insect resistance, easy example. But when I do genome editing, I'm not putting in new DNA. I'm using a mechanism that comes from nature, and I change the DNA, but I change it in a very specific way. So there's only two things that you have to remember of this slide. This is an example, and also you have different technologies through which you can do genome editing. This is CRISPR-Cas, which everybody has heard of, I assume. But CRISPR-Cas has only two things. The big yellow blob is a protein. It's a DNA that can cut DNA. And then very importantly, it's got this piece of guide RNA. So the word guide should already let the bulb come on. Because why do we say genome editing is specific? It's because it has this ability to recognize a very specific sequence. So if you remember anything of matric biology, DNA is a double strand where you have, that A, G, T, and C is the letters of the alphabet, and they complement. A's can only bind with T's, and G's only with C's. So if you have this one sequence here, you need the complement on the other side. It's actually the reverse and the complement. You don't have to worry. All you have to know is it's very specific, that interaction. So if I know gene X, it's got a specific sequence of A, G, T, C, G, and I design this guide RNA of mine, I can actually pinpoint that gene in that cell. And if I attach then with all of these other things that the clever people figured out, if I fix that to the protein, it will go into the cell. Because this protein's job is to cut DNA, I will end up with my chromosome cut into two pieces at that exact spot which I targeted. And then after that, you have two possible mechanisms. The one is just called non-homologous end joining. Forget, it's a ridiculous term in my mind. But the point is, you cut that DNA now with your tool. But DNA can't just stay like this, because then you're going to have so much cancer and problems in your cell. Every cell that divides is going to look weird afterwards. So your cell's got mechanism that fixes DNA the whole time, actively. So all that happens in this case is the mechanisms that's already in the cell, the sugarcane cell or the sunflower cell, or even if you do it on humans, that will fix it. But that fixing process often gives you mutations, a small little error. And it's those errors that's actually the basis of mutation when we talk about evolution. But now we've got a way of introducing them and designing them to introduce it. So we increase the chances, in other words. So we fix the target and we increase the chance of bringing in mutations. Or you can have recombination where you actually insert a gene. So the other thing that you can now remember is that this is why we have those three different categories. And the only reason I bore you with this detail is because it's important when we're going to talk about regulation. And the only point I want to make is SDN1, you just cut the DNA and it's fixed again, and there's some sort of error. You can have one extra base or one is away or whatever, but on SDN3 on the other side, you actually insert a gene. And when I talk about insert a whole new gene, you can start thinking about GMOs again, because this is actually what I'm doing. So you do genome editing, but if you end up with what's on the left-hand side, it's just genome editing. But if you end on the end, because you have done genome editing because you targeted a very specific and you use this mechanism, but you can end up with a GMO. So I know this is complex, but unfortunately, this is the discussions, and this is why we have to understand, because if somebody just comes to you and say, we have to change all the organisms, not change, we have to regulate all the organisms that we've changed over the time. If I don't define that very clearly, it means every animal and plant on your farm needs to be regulated, because they've never been, even the dog that's running around there, because that dog didn't exist hundreds of years ago. It was a wolf or whatever. So you understand this, then we start now have this whole discussion. When is something so different that we have to regulate it? And this is what I'm talking about. Now, I'm first going to talk about the positive stuff, the opportunity. So I'm going to run through these quickly. So what are the opportunities? Why do I get so excited about this technology? So I start off the discussion. I'm going to give you examples, but I always start off with this, because hopefully you will now understand, because I spoke about this genetic variation that's out there in nature. It means actually the potential is as broad as your imagination, what we see in nature. So it's not only limited to maize or soybean, or it can be any organism in the first place, and it can be any trait that we know. So any trait that you can think of in an organism, if it's drought tolerant, if it gives high yields, if it doesn't get infected by a particular bacterium or a fungus, all of those things we can now change potentially. So this is why I start off with this nice one, and it's a little bit bright. I love it. So there's the very first product that was on the market. And now things become complicated, because if we talk about GMOs, it's always regulated, and it has to go through a regulatory process, and then it's put on the market. These first ones also had to go through the process, but through the whole process, most of the regulatory systems in the world decided to say, we're not going to regulate them as GMOs, and I'm going to get to that right at the end. So this is on the market, it's gone through the process, but it's not a regulated product like a GMO is a product. And it's non-browning, very simple. If you eat an apple or whatever, you always see that darkening reaction, especially in some of the older cultivars. Why is that? Cells are broken. When are cells usually broken in nature? It's when an insect eats you. And you have to protect yourself because you're a plant, you can't run away. So you have these chemical warfare things going on there. And this is the polyphenol oxidase. It's a very long, complicated carbohydrate thing that doesn't taste well. So if the insect starts eating it, it will stop. I oversimplify, but that's the basis. And then, I missed the tomatoes. Tomatoes, many plants make vitamin D, but usually at very low concentrations. If they can make vitamin D, it means the mechanisms are there. In other words, the genes are there. But those things are very finely regulated. What we can do now with genetic engineering is to say, let's take away some of that regulation, and you get more, because usually all of these metabolites are products in a very complex metabolic pathway. So it just means one molecule is converted through an enzymatic reaction. That enzyme is a protein that comes from a gene to the next metabolite, and the next one, and the next one. So you have this whole cascade of reactions with different enzymes in it. Usually it's very finely regulated, because as a plant, you think, sugarcane. Sugarcane doesn't want to make all of that sucrose. What is it going to do with it? It's us that bred it to make the sugar, because we want to make, hot, nice, steaming, sweet coffee. But the plant doesn't want that much. He wants just a little bit of sucrose, because it's important sugar in plants, as you know, but it doesn't want to make all these tons that we want to do. So that's an example of how through breeding, we change the plant from accumulating no or very little sugar to a hell of a lot of sugar. And vitamin D is the same story. So those products have already been developed. There's one, gluten, wheat, very complex protein mixture that lots of people have reactions to. I thought it's primarily, a story, but I see people now, and I believe more people now when they tell me they are gluten intolerant. At some stage, it was a very sexy thing. Everybody was intolerant. I'm also intolerant, but not to gluten. Then soybean, I had to put this back in because it at least has some oil link to sunflower. And what they do there is they make it healthier oils. Again, the metabolism and doing it is not that simple, but understanding that you have something that will make you two different types of oils, and it happens with genes and whatever, it's a system. If I understand that system, and that system is based on genetic information, I can change it and hopefully end up with a better product. I tried looking for something in sunflower, and there's nothing because there are bottlenecks. Transformation, there's also no sugar. Sugar, I keep on saying sugar cane because it starts with the S, but sunflower GM, because they can't make, the tissue culture seems to be a huge issue. They can't actually, it's very low efficiencies of transformation. So you see, that must link now down. Suddenly, if you say, oh, this is all this potential and we need to do X, Y, and Z. But there's a very basic research and development question that still first needs to be sorted out. And big players like Innovative, it's a big Bill and Melinda Gates funded organization. And I see actually they say completed project, but there's such a small little summary there that it doesn't seem like they could actually do it. Then we can change track completely to animals. Poenskop. Poenskop cows don't have horns. Why don't poenskop have horns, but the other ones have horns? And if you milk them and do other meat, you do not want horns. So usually when the calf is small, everybody goes and cut them off. It's animal, what do they call it? I don't want to say cruelty because it's not cruelty, but that when things must be happy and, you know, yeah, the ethics is the basis of that. But I can't remember the word I'm looking for, but you actually have to cut it, you know, into the life. We always say, snow lever, because, and then burn it, that it doesn't regrow again. So it's a painful experience. This is where I want to get to. But now it's a single change. So getting back from the, and this is becomes a big thing. We'll talk about stuff again. If you grow up on a farm and you do these things, I mean, you do it every day. I still remember. Cut off the lambs' tails, dogs. Dogs, actually in South Africa, it's not legal anymore. But you grow up with it. This is the big thing of frame of reference. In the end of the day, your product goes somewhere. So if those people do not support what you're doing, you have to adapt. Otherwise, you do not have a product. Poenskop is a gene. You can change it in whatever other thing. I can't think of Arakada, Balva, Brahman, or whatever. You can change that one gene in the Brahman. You're not going to make it a non-Brahman or a GM Brahman. You just change that one gene, and it will be a Brahman that doesn't grow horns. And in humans, so if you do research on genome editing, probably 90%, 95% of all the hits that you're going to get is about humans, because it's a hugely powerful medicine or potential medical applications. These were the first examples. This was already in 2013. Two twins, sisters, that had leukemia, take out some of their marrow, so it's stem cells, change them in the lab to take out the mutation that caused the leukemia, put it back into them. They keep on growing in their bodies, and they were healthy. While I talk about this, the big ethical thing here, that is nothing to me, taking out cells, changing them, putting them back in your... It's a little bit more complicated than drinking Disprin, but it's the same as Disprin, because you make the choice, you only have an impact on your body. But if you go and you say, please take my sperm and select these ones and edit these ones that my kids, look like X, Y, and Z, or they have more muscles than me, or whatever you want it, then you're making decisions for your progeny, for your kids and your kids' kids. So this is the big ethical discussion. We can change things, you can change things, you can make a decision about you. But should we start interfering? Because we can, in effect now, change, evolution. We have the power and the knowledge to direct our own evolution. I've got 15 minutes left. Sorry, I'm just trying to figure out where I am. If you look, then I gave you now a wide spread of different examples. If you then go and look at a detail like maize, which is a huge crop across the world, so there's lots of interest, there's lots of money behind it, then you can also go and have a look at all the different traits. So this gives you an indication of what are those things that you can change, except with what I did. And it's not only about improving the particular product. It can be things like sterility I saw a paper that came out from South African authors, actually, that's talking about haploid development and all those type of things in South Africa. So you can use the tools, again, as a means to an end. It doesn't have to be, I'm going to change this thing to have the final product. It can just be in between. A few graphs just quickly. Again, this is difficult these days now, because 5 years ago when I gave this presentation, I could say, in the USA, in Argentina, and 4 other countries, they looked at these products, and I can list them all because it was those early days. People just started to bring products out, and everybody was asking, do I regulate, do I regulate? In the meantime, all of those regulatory systems have evolved, and they're not regulating it. And countries like America, the United States, and Australia, for example, has decided not to regulate SDN1 at all. So that is like ordinary breeding. So nobody knows what's going on. Nobody knows which products are developed. The best way of trying to do this, there's a European site now, which I list here, where you can go and have a look at the publications that come out. So if all those details are important to you, you can refer to that. All I want to do is, in the first place, is to tell you, look at the activity. Rice, tomato, maize, soybean. So there's a mixture of different things that we see. In the first place, who's active? Chinese. On the rice one, which are the plants that are fairly easy to transform and are amenable to these technologies. This is another reason. Which are the big economic crops. Rice, of course, is a staple in many parts of the world. Soybean, tomato, and all that. I did not find sunflower for the reasons that I said. The other thing that's important, GM, one thing that I have not shared with you yet, is I said South Africa was one of the front runners of commercializing GM crops. But since the middle 1990s, we only commercialized soy, cotton, and maize. 3 crops, and only 3 traits. Of course, there's different genes, but it is only insect resistance, the BT, and herbicide tolerance, and now, just a couple of years, a drought tolerance one as well. So again, I will talk about it later, but this is the problem. Since the 23, we're going for 30 years, more than 30 years, and we only have 3 products. That's ridiculous in terms of innovation, and only a few traits. Now, with this technology, because of the reasons that I discussed, it's easy. There's a big breadth of different traits. And one very important thing that I will come back to later is look at the types. Improved food, feed quality. And then that's the most important, I think, but it's number 1. I think one of the biggest reasons why we still have debates about GMOs is because the end consumer, the person that actually buys the maize meal in Pick n Pay, doesn't get a benefit. There's, of course, very complex economics and, price, all these yields, high yields. Indirectly, they do benefit, but they don't see it. If there was an organic maize meal and a GM maize meal next to each other, they're exactly the same. And they've heard bad stories about the one and not the other one. So why would they choose that one? So I think one of the biggest things, and I'm running ahead of what I should say, but one of the biggest challenges that we have is to develop products that benefit the end consumers. Then we won't have all of these 50-year-old debate about genetics and nonsense. The types, I told you earlier, there's different tools. You don't have to worry about it. The bottom line is CRISPR-Cas, which I showed you, is by far the most popular one, and there's reasons for it. Cheap, quick, and all those type of things. And then SDN1, the types of mutations, SDN1 is the majority. So what I have there is 99. So in the countries where they decided not to regulate those 2, 99% of the new innovative products that come out are not regulated. But South Africa has made a decision in November, August 2021, to say we should regulate these GMOs. So what does it literally mean? Is we're killing 99% of the potential innovation. It doesn't make sense in my mind. Top 10 countries, China, the usual suspects, nothing too interesting. But the question I want to ask is, there's not a single publication when I last looked at this database, not a single one from South Africa. So this is not the farmers and the people in the industry or whoever's fault. This is people that are doing the research. And I understand why. When I talk to people, remember, I'm also academic a couple of years ago, so it's not that I have a knife in for them. But there's no enthusiasm because we don't know what the heck is going to happen. Nobody wants to do applied research which is going to be regulated like GMOs. So not a single paper. Look at the numbers already. And this is more than a year old. So we're falling behind at a rate that we will never be able to catch up, in my opinion. Okay, these are boring ones. The point I just want to make here is the diversity of products at that stage when we still could look at it. And let's just look at one aspect. In South Africa, those 3 products with the different genes, who have commercialized them? The multinationals. Because they've got the deep pockets, because you need to recoup all of that huge amounts of costs, which I will show you later. So it's only no South African innovator will ever commercialize a GMO. And I know that's very broad, but I would be the first person to congratulate because you can't afford it. And it's too complex. So look at this. Suddenly in South Africa, you have only one source. It's multinationals of the product. But look at that. SMMEs. What is this? Almost 70% of the innovation comes from the small companies. And this is job opportunities. These are the things that our government say are priorities that we should do. This is where you get it. And then, they either commercialize it if it's viable as a product in its own right, or, it will go into the bigger pipelines of the multinationals. And here's the reason. To commercialize a single GMO, average of 16 years, and will cost you $1 billion, 1.7 billion rand. I mean, who's going to do that in South Africa? So this is official data. This comes from there. I just want to play it down immediately. We can do it, I think, around 10 million. I can't remember now. It's 10 to 80 million, but at least the order of magnitude cheaper than what it looks for the multinationals. And we're busy, for example, with the South African sugar industry to develop this long process for 13 years or whatever. So we can do it. I think South Africa is typically between 80 and 100 million rands, I think, to develop one line of a GMO, which you then at least can breed into subsequent, but, timelines are not short. So the point I want to make is genome editing is for so many reasons better because the productivity is higher. SMMEs, the role players, because it's cheaper, because it's easier, the role players can be different. And then the diversity, that 8 is at the moment just a thumbs up because it's probably much bigger than that. So regulation, I have to talk about it. So hopefully I'm not going to bore you, but this is where we need people's inputs. If you know somebody in some department, strike up the conversations that we can at least talk about it, because one of the issues that I have is we do not talk about it. So why do we regulate GMOs? Very simple. I think of it from a scientist perspective. I'm regulating. Regulation is about managing risks. So what is the risk on GMOs? Is there a risk? If everybody say you can eat it, you must distinguish between the product that we have and the theoretical risk. It's like getting in a car that the steering doesn't really work and it breaks, and then you go to 140 down the N1. Obviously, that's not safe. But cars, on the other hand, if all of those things are in there and you have a roadworthy car, we do it every day. We take that risk. So you must distinguish between the things. This is the point I want to make. So peanuts, peanuts make a protein. For some reason, it caused some people an allergic reaction. So this poor guy, when he eats a peanut, this is what he looks like. If the same guy eats maize, he's a happy chappy because he's not allergic to maize. So the point why I put in that complex pictures in the middle, that's what a protein looks like if you put it in a computer. So this one he's allergic to, that one he's not allergic to. Obviously, there's not only one protein that you eat, it's hundreds in there, thousands probably. But the point is he's not allergic to that. If there's now some dumb guy in a molecular biologist like me in a lab, and he takes that protein because that protein comes from a gene. I can isolate that gene from peanuts and I can put it into maize. This is what gene technology allows me to do. And now that gene, so what do you think is going to happen if that guy eats it? He's going to be an unhappy guy. And this, in one slide, summarizes the risk of GMOs for food safety. So this is the problem. This is why the scientists that originally did this work in the 1970s got together and said, listen, there's a risk associated with this work. we must regulate it. It's the scientists that said we do it. So it's not this thing, of people having big banners and stuff and say, oh, yeah, we're doing things to nature and whatever. It's the scientists that said, because we understand the risk. Because we understand the risk, we can manage the risk, and that's what regulation. So GMOs introduced this new context of risk, which we have to regulate. So I would be the first person that says GMOs should be regulated, but it doesn't mean that there's now, just like my example of the car. Cars are also regulated. You need a license, all the examples I already gave. But it doesn't mean that we do not have access to cars. We put a system in place to make sure that it's safe enough for most of people to be able to use them. So what has GMOs brought in is this difference between natural risk that we see around us and the risk that we may introduce because we can now engineer things. What is very important is this is not a threshold of acceptability. Because you know of plants in your garden that if you eat them, you're going to die. They're poisonous. So it doesn't mean natural variation or natural risk is always acceptable and good risk. It just means we have a threshold. Above that threshold, we're uncertain. We go through a regulatory process that requires the data, etc., and we regulate it. Okay. Very quickly, we end up with a framework that looks like this, where the GMO Act is the primary one. It looks at health, environment, and socioeconomic aspects. But in the different departments, health obviously looks at the health aspects of GMOs. Department of Forestry, Fisheries, and the Environment look at all the environmental things, and all these acts referred. And then the Consumer Protection Act also came in 2008 and said, yeah, we have to label it. It's a whole lecture on its own. So you see, we have got a complicated one. So now the question is, where I started off, are genome-edited products, or some of them, GMOs? And this, why do I ask this question for you? Is because I want to now give you some information. I want you to think about it. But this has also been the discussion since 2015 in South Africa, is to say, should we regulate them or not? If we regulate them, I hope you've seen clearly that we're going to kill the innovation. And if we regulate it, our assumption is, remember I said regulation is about managing risk. So we say all genome-edited products have a risk, like I just told you, that natural kind of threshold. But you'll also remember that I told you earlier, if I do certain mutations, it doesn't matter if it happens in nature itself, or if I do it through breeding, or if I do it with genome editing. I can't distinguish between it in the end of the day. So how can I say the one that I did in the lab has a higher risk than the one that came from nature? It doesn't make sense in my head because it's the same thing. So we discuss these things ad infinitum. And one of the reasons, and this is one slide of, again, a whole discussion that we can have about acceptability and how people see risk and risk perceptions and all that thing. But if you talk to the average person and you say you're changing DNA, because they've heard, DNA is the blueprint of life, and you're changing life. You understand? I would be, and I'm a biologist, and I still feel a little bit squirmish sometimes when people talk about things. So there's the scary thing out there. And this is what impacts people's perception. And this is one other message that you should remember. It doesn't matter how much I want to convince somebody about the safety of a GMO. If they have a perception which they got from Google, my facts is not going to make a difference because they saw it on Google as well. So it's just two pieces of information that they have to weigh up, and in their perception, the one of least risk is the one is not to buy, for example, a GMO. And this is why we have the debate there. So it's that uncertainty. The point I want to make is a perception. Even if that perception is completely wrong, for that person whose perception it is the truth. It is the frame of reference in which they make the decision. So we need to appreciate and do that. And one of the important points I want to make about this is, like I said, a lot of reasons why we think dread and all. Dread is a thing like, we think Kuba is going to explode. Why don't we like nuclear power, but we like coal? Although coal has us where it has us at the moment, not only environmental issues, but in South Africa as well. But Kuba and nuclear, it has that potential. What we think of, and now I forgot the place name in Russia that exploded, Chernobyl. Obviously, if you compare all of the smog that's hanging around Gauteng, you drive in here, you know, some people know there's a risk, but you're used to it. You drove on it yesterday, the day before, and last week as well. So I'm not going to die of it. But if there's a Kuberg here, and you remember Chernobyl, you think tomorrow. everything's going to be gone. That is what dread is. And biology has that same thing. If we say we're going to change our food, and the food is going to be, how many times have you heard, the tomatoes of today is not as nutritional as the one many years ago. It's a complex argument. There can be some tomatoes that have more nutrition. That's not the point. But this general assertion, that we down the dumps in terms of all our foods and stuff and not. And what is it? It's fueled by this accessibility. I say accessibility to and by audiences. So in the first place, people that want to make money about telling people you're going to die. There's ways of doing that. And there's organizations that make it their business to do that because they will get money to do it. And then the audiences. It's easy. I remember the days that I grew up, you had to go once a week to the library and you get an old book that was published 20 years ago with 30-year-old information, but it was all we had. Now you can go on the internet, you can find out what happened this afternoon in Russia or all the places. So we live in a different world, it's accessible. And the whole discussion, I must finish, I see. Genetically modified organisms, this is the definition in the Act. It says, GMO means an organism, the genes or genetic material of which have been modified. So if I see this, I see there's something in front of me, that example that I gave you of the maize that's been changed. And it's been changed, and there's a risk associated with now. So I have a product focus. But if I change to this one, it's the same definition. All that I did is I highlighted the different part of the definition. It says, modified in a way that does not. So you say, the difference is actually not that gene that's in there that we changed that now may cause an allergic reaction. You say, humans have changed this. And this is this whole story of, because humans have touched it, this commercial farming and all the poisons that they throw on the plants, that is the kind of argument. So you focus on the process. And it's a huge difference between product and process, because if you have a product focus, you can say this thing is different, like I tried to show you with the GMO has now increased risk because of that new gene. But if somebody says it's the process, remember my slide where I had the 3 slides. There it says natural, that mutation can happen, and we do not have to worry about it because it's nature and it's all, wisdom that created that mutation. It should not be regulated. But if those people in the white jackets do it in the lab, we must regulate it because then it's. So you understand, it influences the whole, so the scope of your regulation is completely different. And then just a discussion on theory of risk, likelihood. I'm not going to do this because I said all of these things actually while I did it, so I'm not going to bore you with the detail of my slide. But just on the right-hand side, just quickly, what I also explained is I could have a single process like CRISPR and I can make 2 different products. The one can be indistinguishable from a natural product, or I can use it to make a GMO. Vice versa, I can use either CRISPR-Cas or mutagenesis to create exactly the same one. But some people say the CRISPR-Cas should be regulated, but not the mutagenesis. So it becomes complex. This is what we suggested to the regulator already. This was published in 2015, and I think this is the science. What I wanted to say, I'll finish my sentence, is the next slide. I'm going to show you the timeline. But this is, the only point I want to make is, this is the science behind risk analysis and risk management, ensuring that the product that you can buy is as safe as the natural one. And that's hard science that's been developed over more than 3 decades. So the thinking that the GM product that's on the market, because the difference between GM, the theory of risk, and the product on the market is time, it's detail, it's specifically focused assessment to make sure this product is safe before I do it. You can't just have a theoretical argument. This is the process for genome editing regulations. Specifically, it started here in 2015. Argentina published their regulations. South Africa, the red one there, right on the right-hand side, October 2021, published a 1-pager that says we're going to regulate it as GMOs. AgBiz immediately appealed. That appeal went in. We heard, theoretically, you can see there the appeal board's deadline should have been just, I think it's 3 months plus 3. If they do, They only appointed at the end of the year, so it was about 13 months, if I remember correctly. And the minister has still not made the final decision on that thing. So the difference between 2015, we're sitting in 2023, that concerns me. Especially from a department like agriculture, which must say, in my opinion, we need innovation. We need to support innovation because it's a primary industry in South Africa. All the opportunities, you know all of those stories. I just think there's not a reflection of urgency in this bar, and this is what concerns me. If you look in the world, the green is who said we're not going to regulate it. South Africa and New Zealand are the only two at the moment that said we're going to regulate it. So what do you think is this going to do in terms of trade and all those things? What is it going to do? Remember, my primary responsibility is to support innovation in South Africa, biotech innovation. In agriculture, across everything to health. But in agriculture and the trade and stuff, we're killing ourselves. So my conclusions, why should you care? Because in the end of the day, the basis, the small details that I spoke about can have big impact. The detail is not important, but locally, regionally, and internationally in the world, we cut all of these things in terms of genome editing. You know, it's shorter. You remember my barriers in the beginning? So we cut through it, but now we put another one in and want to regulate it. Doesn't make sense. So this thing, all I said there is we need fit-for-purpose regulation. Fit-for-purpose means we should regulate according to the risk. If somebody dies in a motor accident tomorrow, we can't close all the roads in South Africa. It's a very stupid example, but this is the type of thing that, you know, it must align. And then how do we get it? So this is the important slide I think I just wanted to share with you. We need policies that support explicitly. And we're not doing too bad in South Africa, I must be honest, but it dilutes a little bit when individuals start to interpret it. But the government, I mean, we have commercialized GM crops in 1995. And the problem with policy, it's in a political environment. So it's obviously not only one part that's talking. There are people that have the opposite opinion that I have. And government has to tread carefully, you know, to try and not chase some people away. Regulation, we need a product basis. I explained all of these things. We need to change our regulation in South Africa. Just the words, at least, that it fits. And then RD&I, I think the bottom two are, for me, the important ones to you as an audience, is to say, you will not get a genome edited, all this potential that I talk about that's locked up in genetics in sunflower, if you do not support the research that's going to create the environment in which this innovation is going to happen. And where else is this going to happen? I think industries need to support the things that they want. The days that we sit back, you know, and wait for taxpayers' money to do things like that is probably past. Somebody must take ownership, and there's not ownership at the moment. And then market development and impact, this is the whole thing about make sure what your end consumers want. If you bring up a thing which you can say proudly genetically engineered, and for these reasons, this oil is healthy, it's going to last longer in your fryers, whatever the things that are important to you as sunflower producers. Those are the things that people want. Then they will look at the risk differently because benefit has an impact on how we look at risk. One of the other theories that I have not discussed. So we must be confident, and I don't see confidence in our innovation. You remember I spoke about the people at universities. They're down in the dumps because what am I going to do with my stuff? You know, there's no enthusiasm, and we need enthusiasm, otherwise we're just going to get further and further behind. That's it. Thank you so much.
Thank you very much. I hope that was thought-provoking and stimulating. A lot of work to be done. I really appreciate your presentation.